Um, Hopefully everybody has the report in front of them. That's item 4.2, and it was distributed as an add-on report because the first one, there was some issues with the printing of some figures. So the report content didn't change. It's just some of the figures didn't have their proper labeling. And before I start, is Liz Bowen on the line?
Yes, I'm here.
Awesome. Morning, Liz.
Morning.
So I think Robert hit it on the head. What in the world is transcriptomics? Transcriptomics, that's how I say it. Some people say transcriptomics, potato. I barely understood what transcriptomics was before I started this project or started managing this project. So hopefully by the end of this presentation, you'll have a better idea of what we're doing with the council's money. So the objective of this project is it's a 2-year pilot study to figure out whether or not this doing transcriptomics with mussels in Port Valdez is of value for the council in terms of its long-term environmental monitoring program, which is something that we're supposed to do as part of Open 90. That's one of our key duties. And like I mentioned, on the line is Dr. Elizabeth Bowen. She's an ecologist with the USGS down in Davis, California. And she's done a lot of research, or she's done a lot of transcriptomics work up here in Alaska before. So we decided to work with her and some of her team, who include Katrina Cunahan from the Alaska Sea Life Center, some of you might know, to help us do this work. And after this presentation, I hope you have an understanding of what transcriptomics is. Basically, it's measuring gene expression. I think we all know what genes are in our cells, so that's basically all it is. Number 2, that our long-term environmental monitoring program could use some improvement. It's currently limited. That 3, that this year's results, the first of the 2-year pilot study, have showed that there's some response in the mussels to contamination in the environment that we haven't seen previously, so it's interesting. And with that, transcriptomics is looking like a promising new tool that could be incorporated into the long-term environmental monitoring project if these results keep going on and SAC thinks it's a value and you think it's a value. So why should we be doing this kind of work to begin with? Why are we trying to expand LTEMP potentially? And I think that this quote from Tom Barrett got me thinking about it. So I'll just let you take a minute to read it. Tom Barrett's the Alaska president, the current one. He's going to be replaced pretty soon here, is in transition anyway. But this was a quote from an op-ed piece from Mr. Barrett in the ADN recently. And basically it's saying that, he's talking about hydrocarbon levels, us measuring hydrocarbon levels, and that's basically our long-term environmental monitoring project right now. By measuring that, we're finding that the impacts of Alaska's Prince William Sound operations and tankers are at an all-time low. So I thought that was a really interesting thing to say, and it just got me thinking, you know, as the new project manager for LTEMP, you know, is this conclusion valid? Is what he's saying valid? But because it's a broad conclusion, it's talking about the environmental impact of all of these things happening in the terminal, and is what we're doing really supporting that? So to understand whether or not that's true, you first got to know what LTEMP is. So right now, LTEMP is going over by the Valdez Marine Terminal, dunking this thing into the water and collecting sediment off the bottom and measuring oil pollution in that sediment. And so we do that every year over by berth 4. I think, yeah, we're off berth 4 here. That's Jim Payne, one of our contractors. And we try to measure oil contamination. And the results have been that the concentrations of oil contamination have been trending lower and essentially at this point have flatlined. So do those results support Barrett's conclusion? Somewhat. So they've been lower before, but they are flatlined. So I would say this result somewhat supports Barrett's conclusion about the impacts of the terminal being low. Number 2, LTEMP is getting mussels from Port Valdez. So that's some mussels that we're gathering right next to berth 5 there. And we take these mussels, we put them in a blender, and we measure the oil contamination in their tissues. And what have the results been of that? The results of that in the last 2 years, and you haven't seen the 2019 results, but what we're finding is that the oil contamination in those mussels is so low, the chemistry equipment can't even measure it. So that means things are low. And so does that support Barrett's conclusion? I would say it does. Third, LTEMP is going out by the terminal and other places in Prince William Sound and putting these little plastic strips that I have in my hands there, dumping them in the water column for 30 days, and then measuring the oil that's floating around in the water, essentially. And what have the results been since 2016 when we started this work? The results have shown that oil pollution in the water is very low, lower than what is known to be toxic to marine organisms. So does this support Barrett's conclusion? It does. But just because those three lines of evidence are supportive of that conclusion, I would say that a broad conclusion like that requires broad evidence, and those three lines of evidence aren't enough. So I don't think that LTEMP is currently comprehensive enough. It's only partially supportive of that claim. Right now, I would characterize LTEMP as being more chemistry focused than anything, and I think there's a need to look more at the biology and potentially even some more physical science studies to help bolster the hypothesis that Barrett's making in his opinion piece. And I think it's a great working hypothesis that the environmental impacts of the terminal and tankers are low. There isn't much evidence supporting otherwise at this point, but I think it's our job under LTEMP to bolster that or disprove it. And so where does transcriptomics fit into this? It's a potential tool to help bolster that claim, to prove it or disprove it. So transcriptomics, what in the world is transcriptomics? So we have to understand a little bit of basic biochemistry before you can understand the results of this project. So basically the focus of transcriptomics is on this middle thing right here, RNA, ribonucleic acid. So at the end of the day, what Liz Bowen is measuring down in Davis is the amount of RNA in these muscle tissues that correspond to certain genes, all right? So that's what transcriptomics is in a nutshell. So I'll walk you through this process that I have up here. So in all of our cells, we have DNA, right? And that tells the cell in the body everything you have to do to live. DNA gets turned into RNA. Both are these molecules that are instruction manuals essentially for the body. DNA gets turned into RNA in the cell, and that process is called transcription. Transcriptomics, right? So keep that in mind. The next process is RNA gets turned into proteins. Proteins are little molecules, amino acids that make your body work. If you don't have proteins, your body doesn't work. The process from RNA to proteins is called translation. So now you understand that process. So why do we care? The expression of DNA is affected by two things, both extrinsic factors and intrinsic factors. So what's the difference? Intrinsic factors are like normal everyday stuff. Your red blood cells are constantly dying. They need to be replaced. So genes are turned on in your bones to make more red blood cells. That's extrinsic factors. Your DNA gets turned on to make more red blood cells. That gets turned into RNA. Proteins make more red blood cells for you. Extrinsic factors are things from the outside that affect DNA expression, right? So you get a cold, you get a virus. Once that virus is inside you, DNA inside your cell starts lighting up to make antibodies to fight that virus. It gets expressed into RNA, and then eventually goes into proteins. So how is measuring this transcription telling you something about the environment? So if a mussel is exposed to hot temperatures, its DNA is going to get turned on to deal with that temperature stress, and you're going to see different RNA get produced. If mussels get exposed to disease, certain DNA gets expressed, and then you're going to see certain bits of RNA expressed. So by measuring this amount of RNA that's getting expressed, you're understanding something that's happening in the environment, whether it's disease, oil pollution, or temperature shock. Does everybody understand that? Is that clear? Awesome. So we did this in 2019 in Port Valdez. And this is a map of Port Valdez. Here's the terminal down here. And these little pins are the spots that we took mussels from. So we took mussels from Jackson Point by berth 3. That's one of the LTEMP sites that we usually go to. Saw Island. And then we also took mussels from Gold Creek. That's the LTEMP site's traditional control site. So that we're using that as this is where it's clean. So we're expecting nothing to happen at Gold Creek. And then we also took samples over here from the small boat harbor. And some of you might be wondering, why do we take samples from the boat harbor? And my explanation for that is that mussel transcriptomics is somewhat of a new environmental monitoring tool in terms of its use as environmental monitoring. And we need to understand both what is considered a low value and what is considered a high value so that we can place other samples like Jackson Point and Saw Island in that context. So if Gold Creek's the 0, I would say the Valdez Harbor is going to be the high value in terms of oil pollution, right? Because we're mostly interested in oil pollution here. So that's why we sampled there, is to give context to the results here. So what I like to think of transcriptomics as somewhat is fishing for particular genes, right? So there's particular genes that are associated with oil contamination, temperature, et cetera. And so what we're doing is gathering mussels and trying to see if those genes are there. Just like a fisherman targets a certain species of fish, we're targeting certain species of RNA that are associated with extrinsic outside factors to the mussels. And so what happens is I gathered mussels from the port. I took them out to the lab bench and I did a dissection. I took out their gill tissue. I took out their their gill tissue and their adductor muscle tissue. That's the muscle that opens and closes the muscles. I put them in some tubes and froze it. And then I sent it to Liz Bowen here down in Davis. And Liz and her team down there take all the muscle or that tissue, put it in a blender, and basically isolate the RNA that we're interested in. All right, so they get those pieces of RNA that I mentioned that are associated with certain genes. So what genes did we actually looked at? This is a table of some genes on the left and what impacts their expression. This table isn't inclusive of everything that we looked at in this study. We actually looked at 15 total genes. I'm just giving you these 5 so that you can understand what we were specifically looking at here. So this is just the abbreviation for the genes. They're spelled out in your report. But the take-home message is this gene, CAM, gives you an idea of if pH is stressing the mussels out, if temperature is stressing the mussels out, or if there isn't enough dissolved oxygen in the water column. So if CAM is being expressed, then it's telling you something about these 3 stressors. CASP8, this is a gene that tells you something about whether or not the mussels are getting some kind of disease, or if the mussels are being exposed to oil contamination like polycyclic aromatic hydrocarbons or PAHs. And then finally, you have this MT20 gene. It's a gene in mussels that tells you something about metal contamination. So heavy metals like lead or something like that, if that was having an effect. Now, you might be asking, why in the world are they looking at stuff about pH and temperature? Aren't we supposed to be focused on oil pollution at the Valdez Marine Terminal? It's a great question. The ultimate goal of the LTEMP is to see if the ecosystem is being impacted by the terminal and tankers, right? And that ultimately means that we want to know that these mussels are growing and reproducing within the natural bounds and that it's not being affected by the terminal. But if those mussels aren't growing and reproducing, you need to know why that is. Is it the terminal, or is it something else? And it's a lot better to know what that something else is. Is it pH stress? Is it temperature stress? Than to wave your hands and say, well, it might be something else, but we don't know. So we can't eliminate the terminal as a potential source. So that's why you need to look outside just what you're looking for, so that you can really say with confidence whether or not it's what you're interested in or not. What is going on with this slideshow? Is this just like, is this a challenge, Jennifer?
No, I'm hoping. I'm not touching. I think it's something in the connection.
I was fiddling with it yesterday, like trying to make sure it was all connected, and it is. It's something in the connection, I think.
So it's Nellie's fault. It's okay. If you can turn to page figure 6 in your report, that way it won't be so blinky for you. But this slide is from figure 6 in your report. And basically the take-home message I'd like you to take home from this is that the results in 2019 were promising. Of the 15 genes that we looked at, 5 of those genes showed significant difference between locations that were sampled. And so what that means is that the mussels in Port Valdez were showing an uptick in their gene expression of genes that we're interested in. And those 5 genes were all associated with contamination, either pH contamination or heavy metal contamination. And most of them were the contamination due to PAHs. So that's an important take home. And I'm going to walk through what these graphs mean because this is the bulk of the report and they're hard to understand without some explanation. So first off, this chart is talking about tumor protein 53, and all that specific gene tells you something about contamination, oil contamination. So that's important to know. The y-axis, this is showing the amount of transcription of the gene. And you'll notice that it's at negative numbers and it goes from high to low. That's super weird, right? It should be high numbers up here. The way you interpret this is the higher it is, the more gene transcription is going on. So Valdez Harbor has the most gene transcription and Prince William Sound Field has the least. So it's counterintuitive and it's a factor of how Liz calculates gene transcription. So, but really what you need to know is that the higher, the more transcription. So that's why this arrow is here. And then on the x-axis, you have Prince William Sound Field. These are sites that Liz, in previous research around Knight Island that are clean, that they did this mussel transcriptomics on. Harbor, these are harbors that Liz previously researched with transcriptomics. It's Seward, Cordova, and Whittier. AMT, that's the Alyeska Marine Terminal. That's actually just berth 5. That's that Saw Island point. Jackson Point, that's by berth 3. Gold Creek across the port, and then Valdez Harbor. And then, so what are these red boxes? They're box plots. And the take home here is the little line here is the median value. And this is the interquartile range. And then these lines here at the top and bottom are the 90th and 10th percentile of results. And then these dots are outliers. So you don't really account for, or they're good to know about, but they're not considered really part of the data. So what's the use of a box plot? Basically, if these boxes don't overlap, it tells you that they're different. If they overlap, they're not different. And so what is the story that this graph is telling you, okay? So all of these boxes on the right don't overlap with the Prince William Sound Field. That's the key to reading this map. The Prince William Sound Field are places like Johnson Bay where there is no known sources of PAHs, either from Exxon Valdez, it's clean, right? But places like the harbor, you know there's diesel spills, you know that there's contamination in that harbor. So it makes sense that there's an uptick in that gene expression at that harbor. And what we found in 2019 is that by the terminal, both here and here, over at Gold Creek, and especially at Valdez Harbor, these sites in Port Valdez were more similar to those likely contaminated harbors than to a clean harbor. So that's an interesting result, right? It's saying that Port Valdez is more like a harbor than a pristine site in Prince William Sound. And so that gives you some information of like, what is causing that? Is it the terminal or is it some other source of contamination? And with these results alone, you can't make that determination, right? You need other lines of evidence, such as the chemistry that we're doing with LTEMP. And right now, the chemistry that we're doing with LTEMP actually you can't ascribe these upticks to the terminal because it's below detection limits. But nonetheless, this is a intriguing result because we can't measure this with the chemistry. We're only measuring this with a more biological tool. It's kind of a biochemistry tool, but so it's a useful result that could further our case for the impacts of the terminal. So take homes. I hope that you understand a little bit more about transcriptomics and why it's useful for monitoring. I think LTEMP is currently limited in some ways, and that this type of method could help improve it. Overall, the response that we saw for transcriptomics in Port Valdez was relatively high, and that there may be some impacts associated with the terminal, but we don't have enough supporting evidence to say whether or not that's true. More work needs to be done. And given the usefulness of this data, I think that it's good for the council to continue to support this pilot work. Next year, we have it in the budget under the LTEMP, so I hope you support it, and I hope you accept this report too. So if you have any questions, we'd love to take them.
Bob Shavelson.
I know, Bob, I'm sorry.
Thank you, Austin. That was pretty interesting. Has there been any attempt to correlate the production of RNA with actual concentrations of contaminants?
Not that in this study. So in the new LTEMP report that's going to come out in May, that'll be the first time where we can pair the chemistry, that PAH chemistry, with these results. And like for example, at birth 4 or birth 5, we took both the muscle samples for the transcriptomics and we took the muscle samples for the PAHs. So we could look at those results and see, is the chemistry telling us the terminal, yes or no? And I think it's saying right now the chemistry is saying no, it's the terminal. There's no signal of the terminal, but the muscles are saying something's lighting up this gene. But the chemistry is saying we can't say it's the terminal. So some of that work we are doing it, trying to look at it, but there might be other ways to boost that correlation. I don't know, Liz, do you have anything to add to that?
No, that was excellent. I think, I don't know if part of your question was that what other studies have been done, but it's been done in many other species. There's a great stellar sea lion study and a dolphin study, among others, that have shown correlations between genes and contaminants. So it can be done. I think we're just starting this, so it hasn't been done here yet, but we're hoping to.
And I guess my follow-up question would be, you have the slide that lists the genes, and there's a statement that they're connected to contamination, and any type of transcription and RNA production in there is related to contamination. But how much do we know of that science? That seems like a fairly important conclusion, that there has to be other factors that probably affect the expression of the DNA and other things. So it can't just be one thing, I would doubt.
Yeah, Liz, I think you'd be best suited to address that.
Okay, you're correct. There have been a lot of studies, controlled experimental studies with mussels, looking at the effects of different contaminants, different contaminant levels on different gene and gene expression. So there is a huge body of literature that we can refer to and say, yes, this is normally what happens. And in this case, we've tried to choose genes that are fairly sensitive to just contaminants in this case. There will be other things that affect them, but we have enough genes that we can probably tease that out. But that's a really good question.
And to add on to that, Bob, in some ways, what she's saying too is looking at these genes, it's like this broad category of contamination, right? It could be other contamination, not just PAHs, right? And so that's where I think it's important to think about other projects that the council is doing right now. So in particular, one that we have on the slate for oxygenated polycyclic aromatic hydrocarbons. That's another it's a chemical that results from the ballast water treatment. Well, we need to confirm that, but other studies are showing that these OPAHs have toxicological effects, right? And we have not been measuring those in LTEMP in the port or at the end of the pipe at the ballast water treatment. So maybe it's those chemicals that are causing the uptick in those contamination genes. So it could be other parts of the haystack that we haven't looked at, or it could be PFAS chemicals in the water. I mean, there's other chemical or other contaminants out there that could be causing this. But right now, these results kind of give us a broad idea of the area to look.
It just seems to me that as climate change ensues, temperature is such an important driver in all this stuff. So that's going to have to be correlated in there to understand really what's going on.
And it is. We consider multiple genes for temperature.
Thank you. Steve Lewis?
Yeah, Dr. Bowen, very briefly, if you could, this is obviously new to me. It's way out of my field of expertise, but how many years has this sort of technology been available for research? And you said there's massive amount of data available. Where is that data coming from? Is this something that's only going on in the US? Is this European? Is it worldwide? Just how widespread is this kind of knowledge these days?
Okay, good questions. I started using this technology, I'm going to date myself, but about 20 years ago. And it really started with human medicine and kind of blossomed from there. And it's been used in various wildlife applications for the last 15 or so years. It's been used worldwide, and I would say there are a couple groups that I'm really, really impressed with that have a lot of mussel research going on in Italy. So there are a couple of places in Europe that are very impressive. There's a lot going on in the United States. People are using it the way we're doing it now. They're also using it a slightly different way where they're looking at hundreds of thousands of genes at once. So it's come a long way. It's not a crystal ball, unfortunately, but it is a great tool for giving you some direction for future research and giving you a little bit more definition. Did that answer it? Or if you have
Yeah,
need
no,
further explanation?
that's totally an adequate answer. Thank you. I was just trying to get a handle for basically how well proven out this technology is, and that's kind of fundamental for deciding a level of support for this kind of work.
When you go to your doctor, they now and in the future, they will be using this technology to help figure out what therapeutic approach to take for anything that you might have going on, and particularly cancers.
Robert Beadle?
It's great stuff. Thank you for the presentation, but I'm just curious, just trying to comprehend what do we hope to accomplish with this? Is it that the terminals has some other pollution in the port there, or it's just human interaction with the harbor? I mean, we've got miles of asphalt, other construction and stuff, and it all runs off into, you know, at what point is it an RCAC issue that if we find out it's just humans in the area, what are we going to accomplish, and where are we going to go with it? At what point do we say that it's not, it is or it isn't the tankers or the terminal, it's just dirty people? I mean, who we are. I mean, think about it. I mean, our biggest pollution is, look at these highways out here. We're putting tar on the roads and running them all over, and the runoff. And where are we going? What are we doing?
Where we're going, where I want to go, and I think the right place to go, is to be able to make claims like Admiral Barrett made, basically. It's to say the impacts from the terminal are low, or the impacts from the terminal are medium, or the impacts from the terminal are high. That's the ultimate goal here, is that we gather enough evidence to be able to say, what are the impacts from the terminal? So that we can confidently say Alyeska is doing all they can to limit their environmental impact, which I believe is true. but we need evidence to support that. And we need more evidence than I think that we currently have to make an ironclad case. We can't just go in it with a few lines of evidence to be able to make broad claims like that. We need to look in the corners. We need to look under some rugs, and we haven't been looking under all the rugs right now. And if it is just humans, then we'd like to know if it's just humans, and we can say, well, we're happy to say that it's not the impacts from the terminal are low, and it's something else going on from these, you know, quote, dirty humans, like you said. So that's the goal, is to differentiate between what Alyeska and the tankers are doing versus what everybody else is doing. That's what this is geared toward. And this is a piece of the puzzle. There's no one metric that you can say, yep, no impacts here. You have to look at multiple lines of evidence.
So how long do you anticipate something like this to get a something conclusive to point in a direction?
I think we'll be doing this for as long as the terminal operates, to continually say this is where we're at. Because you also need to understand, if an accident happens, we need to know where the baseline is, right? So if, you know, there's a big spill from the terminal, we need to know what's normal and what's not. So that's also a benefit of this too, is that baseline understanding of once a big spill happens, how did things change? So even when you're getting low results, it doesn't mean it's not valuable. It tells you where, you know, the natural variability, where you're supposed to be.
Okay, thank you.
Iris.
Thank you. Fascinating. Could you go to the next slide? So my question is, I've been reading a lot more about how stressors can be cross-generational and impact DNA. So is there a possibility or likelihood or way of checking? You know, I look at all those boxes, the overlap in Port Valdez. Could this have been from a previous stressor and it's just propagating forward?
Liz?
Yeah, that's a great question. I think you're talking about DNA methylation events that
Right.
can happen and are inherited. I would say there's a slight chance, but these are more indicative of something that's happening currently.
More acute?
You can check for DNA methylation. It's a different technique.
Okay.
And I know one of William Driscoll, who is also participating in this project, is very interested in looking at DNA methylation to see how much has happened over time and how much of the methylation events have been inherited. But this, in answer to your question, is more of a current occurrence.
Thank you. That just is a personal question. This is a fascinating report. I got to say, I'm kind of tempted to say I'm against it, so the council will vote unanimously to support it going forward.
That's a good one. Donna Shuggs.
Not so much a question or comment on the report, more on some of the information in the presentation. And Austin, you know, you did a great job taking a very complex topic and boiling it down so that we could all understand it. So, you know, really good job. But one thing I know, I talked to Austin about this earlier, and I just wanted to clarify that the quote of Tom Barrett's wasn't necessarily his hypothesis. It was a finding of our last LTEMP report, and we actually did a press release. He may have changed a couple of the words, but we did a press release, you know, with that report, announcing the findings of that report with a very similar statement. So I just wanted to clarify that. He really did pick that up from the findings of our last LTEMP report. So thanks.
Marco Haggerty.
Yeah, I really appreciate the report too. It says in here, and maybe even mentioned it, that this came from one sampling in June. Would it change any because mussels, you know, metabolize fast. And I know with the paralytic shellfish poisoning, you know, they're the first to get it and the first to get rid of it. But this is, you know, you're talking genetic structure here, so that's probably why just one sampling will answer a lot of questions. But is it necessary to like pick it up in the, take several samplings over the course of the year? Would that sharpen this at all, or?
Yeah, I mean, it absolutely would sharpen it. So a great example, and I think actually Wayne brought this up in one of our SAC meetings, was that if you sampled, say, in January, when more ballast water is being offloaded and processed, you're going to have more PAHs in the water at that time from the ballast water treatment facilities, potentially. And so if you sampled in January, you might get a different uptick than you would in June when the tankers aren't carrying as much ballast, and so there's not going to be as much process through the ballast water treatment. So looking across the year would add some benefit to see how things change, because this gene expression is happening all the time. So it would add value to do more. We're not planning on doing it yet because of the pilot study, but it's under consideration and it would be useful.
Dorothy Moore.
Let Jim go. I was ready to move to.
Jim.
Thanks for the report. This kind of builds on the two questions that Andres and Mako just asked. How quickly do these genes turn on and off? How quickly, similarly, do the mussels purge themselves if there is an event? This kind of, like Mako says, if you had an oil spill today, are these genes all going to be putting out RNA for the next 3 months, or is it going to be over in a week? How sensitive, how acute is the response? Compare that to the mussels, either Dr. Bowen or...
Yeah, we're considering that question, but I would defer it to Liz. Yeah.
It's the genes are very sensitive, and I would say relatively, their responses are relatively ephemeral in comparison with some of the other tools and techniques. So if you're exposed to oil today, you would initiate a response, or your genes would initiate a response, which would wane over, about a week period, depending on which particular gene. However, if you're continually exposed to the oil, then you would continually have a response. Does that answer your question all right?
Yes, correct. That's kind of what I surmised. And then that ties back to the overall LTEMP mission. By sampling mussels, you're basically trying to find continued presence of oil as opposed to just a spike. Because if there's not too much in the water, they'll purge themselves and they'll look pretty clean. So...
Yeah, I mean, by and large, yes. But we've also used mussels for acute stuff. So when they had the September spill in 2017, I believe, I went out and we went and sampled mussels. So you can use it for both chronic and acute. So,
John Kinnish.
Dr. Bowen, I have a question about the technique. It's not clear to me about your temperature requirement here. If you're collecting samples and then freezing them and transporting around, is it possible that you could have historical samples evaluated in the same way? So for example, I know that there are samples stored around the country from the Exxon Valdez spill. They could be, some of them I think are stored at -80 degrees. Could those samples be evaluated and compared to what we're seeing today? Because under those conditions, there would have been severe contamination.
That's a great question, and that's great information as well. If samples were stored properly at -80 degrees or in liquid nitrogen, then the RNA should still be viable. When we transport our samples, when we're out in the field, out in Katmai or something, and we don't have access to a whole lot of liquid nitrogen, we put our samples in a special buffer that stops all RNA transcription, so it stabilizes everything. So either or, if a sample has been stored properly at -80, we can still look at it. If things have gone through several freeze thaws, as they often do in freezers when power goes down, etc., then it won't be viable anymore. But those samples that you're talking about would be extremely valuable.
Thank you for that. Andres Morales.
So, we have several different places here. I was wondering if we could actually get some more representative sample from Italy, or, are they genetically too different? I'd like to just see, so how does Prince William Sound stack up against California? Or, that's just, again, interest.
Well, we have, it's a different species of mussel that we've looked at in California, but I've got some data for a beach down in Point Reyes, and I can easily share that with you guys so you can see how it stacks up.
Yeah, for sure.
Any further questions? Yes, Wayne Donaldson.
I just wanted to mention that this was just a feasibility study, the way I understand it. There's been no connection made to the terminal. It's just, we took some mussels from various locations and measured the expression of these 15 genes. So it's just to see whether this is feasible to continue on. There's been no direct link, and there's no long-term commitment to include this in LTEMP unless we get to that point where we can identify hydrocarbons that are causing genes to turn on above the background level. That's my understanding.
Any other questions? Well, this seems like a new and developing technology that is going to be with us in the whole realm of diagnosing things, even in the human body. Interesting. So with that, this is an action item to approve this report.
I would move to approve this report as stated in our book, because I don't have it right in front of me. But I would also like to say, sitting on the SAC committee, I think I've seen this report at least 3 times, and every time I learn something new on it. And Austin, I have to give you kudos. This was an excellent, bring it down to so the person on the street could understand. Thank you.
You're welcome.
So I move.
That was Dorothy Moore first. Who seconded? Robert Beadle. Are there any objections to accepting this report? Very well. Thank you, Austin. Very well done.
You're welcome.